Lecture 5 is going to cover the nervous system. So this is going to be chapter 11 in your textbook. The nervous system is divided into has two components to it. There's the central nervous system, which includes the brain and the spinal cord. And then there's the peripheral nervous system that includes cranial nerves, spinal nerves, and ganglia, which are just a cluster of cell bodies in the peripheral nervous system. And then the peripheral nervous system, that's further divided into different divisions. So there's the sensory division. This is pro in the sensory there's the sensory division. So this is where the afferent stimuli, afferent. So these are signals that are arriving at the spinal cord. So these are detected by neurons, and they're involved in things like gustation, which is taste, audition, which is hearing, somatosensory, touch, and olfaction, which is the sense of smell, and also vision. The visceral component of this, visceral meaning organs, so the visceral stimuli are typically signals from the organs and glands. And then there's the motor division that conducts the motor nerve fibers. So these are impulses that are coming from the central nervous system, and then it's going to the effectors. The somatic uh, division that's involved in the skeletal muscle, so it allows for skeletal muscle movement. The autonomic division is involved in involuntary uh, movement, so for movement of cardiac muscle, smooth muscle, and glands. And then that's further divided into sympathetic division, which is the division that allows for fight or flight, and then the parasympathetic division that allows for rest and digest. Neurons are structural units of the nervous system. So a neuron is just a nerve cell. So these are large and specialized cells that conduct the impulses. They have special characteristics like extreme longevity because a neuron could last a person's lifetime if undamaged. They're amitotic, meaning that they don't divide through mitosis. They have high metabolic rate, which means that they consume a lot of energy and oxygen. And all have a cell body with one or more processes. Okay. So when we look at the structure of a motor neuron, so we can see the nucleus here. The nucleus is going to be surrounded by the cell body. So that's also called the soma. And then coming off of the soma, there's those processes that are called the dendrites. Then there's a long process that's called the axon, and that originates here in this cone-shaped area that's called the axon hillock. The axon, so this is where the action potentials occur, and we're going to go over that again in this video. And you can see that it's on the axon, these are wrapped in these structure that are called the Schwann cells. And this, in between those Schwann cells, you can see that there's parts where they're exposed. That's the area where the voltage gated ion channels are located. So the signal travels in this direction from the cell body to the through the axon, and then it's going to terminate at the axon terminals. And these are the ones that connect to other neurons. So they'll connect to either the dendrites of another neuron or the cell body of another neuron for the most part. There are some exceptions, but typically that's how it happens. They connect to either the dendrites or the cell body of another neuron. The structure of a neuron has only one axon that starts at the axon hillock. And in some neurons, the axons are short or absent, and in others, the axon makes up pretty much the entire length of the cell. So some of the axons can be up to a meter long, which is around 3 feet. The long axons are called nerve fibers, And then the axons branch profusely at the end. So those are the axon terminals here. Okay. And the axon terminals are also called the terminal boutons. So the axon is the conducting region of the neuron. It generates the nerve impulses that transmit along the axonal lemma. So it's just the neuron cell membrane. And that signal travels to the axon terminal, where, like I said, it'll connect to the either the dendrites or the cell body of another neuron. So at the terminal region, that's going to secrete the neurotransmitters, which are going to be released into the extracellular space. And these can either excite or inhibit the neurons it contacts. The myelin sheaths are the structures that are parts of the Schwann cells that are made of myelin, which is a whitish protein lipid substance. And the function of myelin is essentially to protect and to insulate the axon. And when it does that, it increases the speed of the nerve impulse transmission. The myelin sheath gaps, which are between the adjacent Schwann cells, are also known as the nodes of Ranvier. The non-myelinated fibers, these are fibers that are not wrapped in myelin. There's still Schwann cells there, but they're not coiling around the axon. So here you can see the Schwann cells, uh, which again is not the whole blue structure here. It's just a small component of it. But those do contain the essentially the myelin, and that's what's wrapping around the axons. And again, the main function is to protect and electrically insulate the axon to increase the speed of the nerve impulse. On the membrane, there's going to be proteins that are going to serve as ion channels. These large proteins are going to make the cell membrane selective in the type of ion that it allows to cross over. So for example, the potassium channels are only going to allow for potassium to cross. The sodium channels are only going to allow for sodium to cross. So these chemically gated or ligand gated channels are going to only open when binding to a specific chemical, for example, a neurotransmitter. The voltage gated channels are going to open and close in response to the membrane potential. And then the mechanically gated channels open and close in response to physical stimulus. So stimulating those receptors, like the sensory receptors. So here you can see the chemically gated ion channels. These are the ones that open in response to the binding of the neurotransmitter or the appropriate neurotransmitter. So this is just a chemical that sends signals. So once the neurotransmitter binds to the receptor of the channel, then that channel will open and that'll allow for the ions to move. For voltage gated channels, these open in response to changes in the membrane potential. So changes in the charges of the membrane. So if the charges start by being negative, if there's sodium already coming in through other channels, that'll change the charge from negative to positive, okay? And that'll also open these voltage gated channels. Okay. So some basics on electricity. So these ions, so there's going to be different charged ions inside and outside of the membrane. And opposite charges are going to be attracted to each other. So if there's negative ions inside of the cell membrane and positively charged ions outside the cell membrane, those positive charges are going to be attracted to the negative charges, and the negative charges are going to be attracted to the positive charges because the opposite charges are attracted to each other. And also, energy is going to be required to keep those charges separated across a membrane. And when the opposite charges are separated by the um, by the membrane, that system is said to have a potential energy. So here they're measuring the membrane potential in neurons by using an electrode, and it's measuring a negative 70 millivolts. Okay, because it's inside of the membrane is currently more negative. Generating a resting membrane potential is going to depend on two things. It's going to depend on the differences in potassium and sodium concentration inside and outside of the cells. And also, it's going to depend on the differences in permeability um, of these ions. So potassium is always going to be higher inside of the cell. Sodium is always going to be higher outside of the cell. Okay? That's why sodium moves, when it moves down its concentration gradient, it goes into the cell because it's always higher outside. Potassium is always higher inside, so when it moves down the concentration gradient or along the concentration gradient, it always moves outside. And then there's the sodium and potassium pumps, which are going to maintain that concentration gradient of the two ions. So more potassium is going to diffuse out of the cell than sodium is going to diffuse in. So that's why the inside of the cell is going to be more negative. And that's the reason that potassium is the one that establishes the resting membrane potential. The sodium and potassium pump is going to stabilize the resting membrane potential, and it's going to maintain the concentration gradients for sodium and potassium. Because for every 3 sodium ions that are pumped out of the cell, there's going to be 2 potassium ions that are pumped back into the cell. Depolarization of the cell membrane, that's caused by the membrane potential moving closer to 0 millivolts. So that happens when, for example, if sodium, which is a positively charged ion, moves into the cell, those sodium ions are going to cause the inside of the cell to become less negative. So that'll bring the voltage closer to 0. Hyperpolarization is when the membrane potential increases, so the inside becomes more negative. And that happens when potassium, which is a positively charged ion, leaves the cell. That's going to make the inside of the cell more negative. So that'll uh, bring the voltage farther away from 0, right? Like at around negative 90. The principal way that neurons send signals are action potentials, and these only occur in muscle cells and the axons of neurons. So it's a brief reversal of membrane potential with a change in voltage of around 100 millivolts. Action potentials don't decay over distance like graded potentials do. And in neurons, they're also referred to as nerve impulses. And these nerve impulses are going to involve opening of specific voltage gated channels. So here we can see the steps that are involved in the action potentials. So if we start here, okay, right before the action potential, that's considered to be the resting state. That's when all the sodium and potassium gated channels are closed. Depolarization is going to occur when the sodium channels are opened, and it's going to allow for sodium to move down its concentration gradient. So going from an area of high sodium concentration to an area of low sodium concentration. Because remember, sodium is always higher, has a higher concentration outside of the cell. Potassium always has a higher concentration inside of the cell. So when the sodium channel opens, the sodium moves down its concentration gradient or along its concentration gradient. That's going to cause the cell to depolarize because sodium is going in, it's going to make the inside less negative. So that's called depolarization. Because remember, when you have uh, different charges, that's called polarization. But when you make one of those charges, like for example, if the inside is more negative, outside is positive, that would be polarized. But if you make the inside less negative, now you're depolarizing it. So that's why it's called depolarization. And that's that initial part of the action potential here. So after sodium opens, and then eventually those sodium channels are going to close, and as those sodium channels are inactivating, the potassium channels are going to open, and that's going to allow for potassium to move along its concentration gradient. So now those potassium ion voltage gated ion channels are going to be open. Potassium moves along its concentration gradient, so it'll leave the cell. And when it does that, it's going to make the inside of the cell membrane um, more negative. So that's what causes this portion of the action potential. So it goes down. So it makes it more negative. So this is going down, or the voltage is going down. Hyperpolarization occurs because some of those potassium channels are going to remain open, and the sodium channels are going to have to reset. So if those potassium channels remain open, more sodium is still going out, and that's going to continue to make the inside of the cell more negative. So that'll that'll make this it'll make the voltage go lower than it was at resting membrane potential. Okay, so just to recap, we have a resting membrane potential here where no ions are moving through those channels. When the potassium channels open, when the sodium channels open, that means that sodium is going to move down its concentration gradient. It's going to enter the cell, which is going to make the cell less negative. So it'll bring that voltage up closer to zero. Okay. Then those sodium channels are eventually going to start inactivating, and then the potassium channels are going to open. When potassium channels open, that's going to that's going to allow for potassium to leave the cell, because potassium is higher has a higher concentration inside of the cell than outside of the cell. And when it does that, it's going to make the inside of the cell more negative. So it'll bring that charge, or the voltage, down farther away from zero. So that's called repolarization. Hyperpolarization occurs because some of those potassium channels are going to remain open, and they're going to continue to make the inside of the cell more negative. So that's hyperpolarization. Repolarization resets the electrical conditions, as in bringing that charge uh, back to being more negative, but it's not going to reset the ionic conditions. So ionic meaning the the movement of the ions. So after repolarization, the sodium and potassium pumps are going to be the ones that restore the ionic conditions. Okay. The way that the sodium and potassium pump works is that it takes 3 sodium ions and it moves them back out into the cell, moves them back out of the cell, and it brings 2 sodium ions back into the cell. Because remember, so when sodium concentration starts higher outside of the cell, and then when those sodium channels open, it moves into the cell. Potassium starts higher inside of the cell, and when those potassium channels open, potassium leaves the cell. And then to balance, or to bring that back into balance, the sodium and potassium pump will take the sodium that came in, it'll take 3 of those and push them out, and it'll take 2 of the potassium ions that left the cell, and it'll bring them back in. Not all depolarization events are going to produce action potentials. For an action potential to fire, the depolarization must reach a threshold to trigger the action potential. So at threshold, the membrane is depolarized by 15 to 20 millivolts. So that'll be around negative 50 millivolts. Because if the resting membrane potential is 70, then depolarizing it by 15 would make it negative 55 millivolts. So when it reaches a threshold, the sodium permeability is going to increase, and that sodium influx is going to exceed the potassium efflux. So this is going to make the inside more positive, and that's going to create a positive feedback, because as more sodium comes in, the more the inside of the cell membrane is going to depolarize, and that'll open more sodium channels to allow for more sodium to come in. So these action potentials are all or none because they either happen or they don't. Okay? But they have to reach that threshold of negative 55 millivolts. Propagation allows for the action potential to be transmitted from the origin down the axon and then towards the terminals. And the sodium influx through the voltage gated ion channels in one membrane area is going to cause the local currents, and that's going to cause openings of sodium channel gates in the adjacent membrane areas. So this is going to lead to depolarization of that area. Once initiated, the action potential is going to be self-propagating, meaning that it's going to move on its own.